Hello!
Hi
Well,
there.
welcome to YouTube Microbiology Journal Club, where we talk nerd up about all things great that are small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, MIC, dedicated to naming the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I mostly work on bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today it's our news week where we do an overview of papers that we've seen in the news or other sources to try to find an article that we want to cover for the next week in more detail.
And next week will be our deep dive week, where we take an article we choose today and look at it figure by figure to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
So you can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments to tweet us. And without further ado, I think that brings us to this paper. This week, we're going to be focusing on a comment that was raised to us about a paper that we covered, but not really in much detail. And there
Yeah,
turns out there was a much bigger story there that we missed out on.
absolutely. Yeah, no, it was really awesome to get a comment like that. Just somebody else, I guess, who's following along with the literature, they linked to like a bunch of HHMI talks that I
Yeah,
guess a lot of institutions are doing their COVID talks online now. So, search YouTube. I mean, people probably have been searching YouTube to find those things. And yeah, they picked out this paper, the Christian Gabler paper, Evolution of Antibody Immunity to SARS-CoV-2. Um, yeah, I think when we talked about it, we sort of just said, oh, this is like the evolving immune response, right? Like B cells change, you get different antibodies over time. But actually, they say something a little bit, they have an interesting observation where they look at the gut specifically.
yeah. So I'm just going to scroll down to the page because
Hmm.
one of their collaborators found that after I think 92 days, they looked at people who had resolved COVID infection, and they
Hmm.
found that there was some SARS N protein being expressed in gut enterocytes.
Right. Yeah, so yeah, that's kind of That's interesting because that's a long time post-infection, right? Like you've cleared, right? Like you've cleared your symptoms by then, and you're probably considered non-infectious at that point. So it's odd to see that there's something in the gut that's growing. Well, I guess they don't know that it's growing, right? Like this is just this particular protein is there, and so one of the counter arguments right to that is that maybe the protein gets sampled in some way, right? Like it was in your gut, it passed through, right? And then like I don't know, your cells retained it in some way. But I think that's considered unlikely because usually the body will degrade proteins, right? Like it only keeps proteins around if it needs them. So I think the thought here is that there might be active replication.
Yeah, well, I mean, this paper is actually quite careful in how they phrase it because that's part of the reason why I missed it the first time around because they do mention it solely in terms of antigen, and it is true there are some parts of the immune system that do hold on to antigen a lot more, like the dendritic cells that present antigens. Um, so there is the suggestion that maybe the gut cells are doing something odd like that, but then
Yep.
again, if we go into the bioRxiv preprint, we can see a previous version of this article where they actually have some uh, electron microscopy where they find that they have. Let me scroll down to see where. Uh, oh, another kind of note is that they didn't just find it in one patient. They found this in I think 3 out of the 30% of the patients they had.
Hmm.
Um, and let's see,
Yeah,
because I
so there's
think
that photo with the IF again that you just passed.
yeah, I just passed. I'm running through this because they had some. I guess it might be in the sub extended data and legends, which
Definitely.
which is
There's so many of
really
them.
there's many of them. There's a lot in this paper. There's a lot of interesting things in this paper. So this gut is so this partly also like kind of explains another finding that they found in this paper, which oh god I. So this is electron microscopy. That I can't point to specific parts on it because this website is
Without
screwed
wrinkle.
up. But but yeah,
Is
on
that electron microscopy? It doesn't look like electron
I
microscopy to me.
I don't know. Yeah, let me
Let me
see if
see.
I need
I
to. Okay.
have it up too, so I can. I can scroll down to the extended figures. I mean, this is you can put your mouse over it and see what the caption says.
Yeah. Okay. What the caption says is, uh, oh, this is yeah. You're right. This is an electron microscopy. It's at EPCAM. Um,
Mhm. Yeah, so
so
this is more IF actually.
yeah, more IF. Okay, so
More IF.
so during the HMI talk, they did show some electron microscopy of
Mm,
SARS-CoV of well, not of a virus that they found in there that looked very similar to a coronavirus, but the
mhm.
thing about electron microscopy is it gives you lots of structural information, not necessarily genetic information. So I think it might have been from a different paper that we're going to cover today. Um, but
Yeah, I
yeah,
think I mean like doing more certainly doing more IF, right? Like gives you more confidence. They're just showing more pictures, right? More stuff that they've seen. I think that they in this IF they are the EPCAM, that's a marker for cell type, right? So
yes.
they're saying that it's in enterocytes. So that's some information that comes from this as well. Um,
Yes. Uh, yeah.
and yeah,
Um,
and I think they also look along the whole. It looks like they're looking along the whole like all the sections of the intestine too, right? Like
yes.
they have colon, um, terminal ileum, duodenum, and they're seeing these like little foci of nuclear protein, nucleocapsid.
Yeah. And part of this is quite nice, but also quite isn't very helpful because ideally you'd want it to be a specific area of the colon so you know where to target when you're looking searching for more. But having it, and the thing is, colon is a very difficult place to sample because first you have to find a patient who's willing to let you take a biopsy, and then you have to
Yeah.
and you only get that what's in that biopsy. And colon is and the intestinal system is a big system, and there is a
Mhm.
chance that you might miss it as well. So the one that's one of the things that came up in this talk where that they're saying, okay, well they sample these, but they weren't looking everywhere in the colon. It could be in other regions. So
Yep.
uh, yeah, so that's another difficult conversation to have.
But yeah, I mean, it this is the hints, right, of like, oh, what's going on in the in the gut here with SARS-CoV-2, and especially at this late time point, I think is what is curious.
Yeah, and another bit of evidence, line of evidence that they had. I think the thing is, this was a good thing to explain something else that they found during their study. So, uh, what they found was that the neutralizing activity of these like antibodies to SARS-CoV-2 did develop during the
Mhm.
infection. So, so what they found was that from they pulled out some antibodies from
Mhm.
each like kind of section. So uh, they pulled out antibodies from like the initial stage where the initial infection was going, and they tested whether those would uh, neutralize antibody escape mutants. And then
Mhm.
they sampled antibodies taken 92 days afterwards and sampled whether they could neutralize the antibody escape mutants. And weirdly, the ones at that later time point did neutralize those escape mutants, implying that
Yeah.
the immune system evolved its response to SARS-CoV-2. But
Yep.
and this is where the confusion comes in because how does the immune system know that this mutation has happened? And
Right.
the conclusions that these authors have kind of come to is perhaps I it's because they're so, whether through a dormant infection, whether it's a long term infection, or even if it's just holding on to these antigens, that the immune system is is using this of evolution of sorry, I'm getting
Yeah.
my words are but using these antigens to develop its immune response. And
Right.
one of the only like yeah one of the ways to kind of rationalize this is saying that okay, there is a long-term dormant SARS-CoV-2 infection that's happening that is being used to develop the immune system. Or
Being used. I don't
that's
know, like
yeah,
used by what?
you're right. But it's sorry, it's
um.
causing the immune system to evolve. Is probably a better way to phrase
Right.
that.
Mhm.
um,
Yeah, that is interesting because I think time scale is like really important in this particular discussion, right? Because we know, and we've said this before, that there is an evolution, right, in B cells when upon exposure to antigen, right?
mm,
And that exposure is supposed to make the antibodies bind better to the specific antigen. But what's strange about this finding is that it's happening a lot longer,
Mm,
and it's no longer recognizing what we would think of as the antigen, but rather this variant antigen, right?
mm.
With these point mutations. So I guess the suggestion would be that inside of this patient there is the evolution of the variant, and then that variant is being recognized by the immune system, and an appropriate immune response is being mounted, and that happens quite quite a long time after, right?
Yeah.
One would think the infection should be over.
So yeah, it's quite a big thing, and so yeah, it's so I imagine that can be a step too far for some people, perhaps, because I mean it seems very extraordinary, and so it's something that we'd like to delve in a bit deeper to see whether there's any other work in this area,
Yes, yes, absolutely.
which is what we've been doing for the past week. So we've been
Yes. So that gives us some stuff to talk about, some more things. Oh, I guess the first thing we'll jump to is uh, just what's known about the interplay between gut and respiratory infections. And so this is a review,
yep.
and it's published like before. Oh, and I guess it is published after.
It's
uh,
published after, but it was probably written like before. Because,
Yeah, before. It wasn't, it's not written like with the thought of SARS-CoV-2 necessarily,
hmm,
but rather um, the interconnectedness of our body and infections in different locations. So if we can go down to maybe figure one, I think that's the best overview. And so if you think about it, like the gut and the the gut and the lung are like totally separate areas. They have their own microbiota, they have their own things going on, but they're connected because the blood connects those two systems, right? They're connected through our immune system. And I
hmm,
think the biggest point to take away from this is that you can have immune responses in one place in your body, right? Consider the gut, right? Or like let's say the primary infection in the lung, we'll say,
hmm.
and that's going to template how the body reacts to things that come through the gut. So I think in this discussion, right, the point to bring in is that you could have this respiratory infection that sets your body up to recognize these pathogens in some way. Then you could ingest those pathogens, and the response that you would get is different, right? The body doesn't want to over respond to things, right? And in responding to things, it could be driving the evolution of the virus, right, in that location. So like these are just the ways, I guess, in which it could happen. There's nothing definitive in this paper to really speak to what's going on. The just to highlight, I think what I wanted, why I brought this paper up is I just want to highlight that interconnectedness. That it's connected by blood.
Yeah, and they are technically, I guess, part of the same tube as well, somewhat.
I
And
guess they split off right at the epiglottis.
but yeah, and I think there has been this sense of of like kind of linkageness, the idea that if you vaccinate one area, so if you take an intranasal vaccine, that can also have impacts on the lungs, or perhaps even have impacts on the gut.
Yes.
So they are linked because I think the methods in which the immune system work is very similar between different areas of the mucosa. So there is that unified.
Well, I mean, I think that that's an interest. Like, we haven't really talked about like the immune system in this level of detail, but there are immune cells that sit in tissue versus immune cells that flow in the blood.
Yeah.
And we've talked a lot about this idea of in some of the past episodes we've had, we've talked about circulating immune cells going into the lymph system and being trained there.
Mm.
But like there are also then feedback to the resident immune cells in locations, right? Like I mean, there's all sorts of very complicated sort of stories, I think, that are told about these enzymes, but what makes an immune cell decide that, oh, it's time to move into the lymph system? What makes an immune system go back to the tissue that it was resident in and maybe inform the other cells in that area what's going on? There's lots of stuff like that's going on, and it has to communicate through blood. So there's all sorts of mechanisms in the immune system that allow those types that information to flow. And yeah, it sort of stands to reason that it should happen in this infection as well. Just what's the actual impact? That's that we can't. That's not what's being studied here. This is a review about sort of more general themes, I guess you could say, right? That the immune system does.
Yeah, so a review is a good way to get into the field if you're not entirely familiar with it. So usually they're written they don't present real kind of original research, but they do give you an overview of what's happening in the field.
Yeah, and
So
they'll link to the original research, right? Of all these different things
yes,
that support the type of interpretation that they're bringing to the table.
yeah. So if there's something you find in the read that you particularly find interesting, just go to the reference list and just drill down. There's so many links now these days. Eh, we're so far from the olden days where you have to like ask a librarian politely and then a week or so later you get a paper. No, this is an era of the internet, so it's so good.
And we're kind of like
And
a casual form of a review, right? Like these reviews, like people sit down and they think very hard about like all the different references to bring together. We're sort of doing a casual version of a review in these episodes, right? Where we just
oh
grab
yeah,
a
I mean
smattering of things and mash them together.
yeah, I mean I would say that we are in no way like as thorough as many reviews are. I mean, but we do try our best to give a little summary of papers where. Almost like an email chain that you might get where someone goes, "Oh, here's some nice papers you might like to read." That's kind
Yeah.
of us.
I think the other thing that's we haven't really looked at a review like in this session, so I just want to highlight for people that reviews are also like written by people in the field. So like it's when people get really focused on a certain topic, right? Like they had to have read all of these papers in order to be working where they work, and this is a way of catching people up to that.
Yeah.
Mhm.
Uh, so next paper is evidence for gastrointestinal infection of SARS-CoV-2. So this was one of the first papers that came out about this topic. So it was published in March 2020, and so again, this was just where they started to
Yeah,
just
that's like right at the beginning.
right at the beginning, a brief communication where they were literally saying, "Oh, look at this! We found something new about this new disease. Please
Mhm.
let us tell you about this." And then they were doing also histological images standing for the same I believe it's the same protein that oh,
The
uh,
nucleocapsid.
yeah, the
Uh,
the N protein, I believe.
yeah, it says
Uh,
E. Yeah, nuclear protein.
nuclear protein. Yeah, yes.
And I think the other thing that's nice about that figure is that they stain for ACE2. So they're also linking that, right? Like ACE2 is expressed on cells in the gut, and we know
Mm.
that the ACE2 is the receptor. So it's giving us that little bit of information, a confirmation, right? It's not crazy to think that they're
So
infecting the gut.
yeah. So yeah, about like a year ago, this was kind of considered sufficient evidence that there was something in the gut, I guess, for gastroenterology at least. Different journals have different thresholds for what they're willing to publish. But
Yeah,
yeah,
it's a pretty small report. Though they look in a couple, right? They're looking at a couple human patients under
yeah,
25 male, 14 female, they say.
yeah. It's not very big. It's a brief communication, so this is literally done to be as quick as possible. So they're usually very short, very much like here's something that everyone needs to know immediately. Uh, and then the next paper is a prolonged presence of viral RNA in gut samples. Um,
Mhm. In fecal
in fecal
samples.
samples, sorry. Yeah. Oh, where's my head today?
It comes from the gut.
It comes from the gut, but sounds about specificity, and I'm keeping getting it wrong today. Um, uh, but this is another paper looking at like how long of RNA can be found in fecal samples, and what they
Yes.
found was quite interesting because they found that it can actually be quite a long, about like over the time maybe a month or so, so
Hmm.
quite a while after the original infection has been resolved.
Yeah.
Um,
Which is again a strange. It's a strange observation if what we're thinking is, oh, during your primary infection is when you have replication in all these parts of your body, right? And then after that, everything just gets cleared out, right? Your body churns over. But to have such a long period of time, right, to still detect that viral RNA, I think this is part of why we can say it is trying to support the idea of some sort of replication going on.
Yeah, again, this is a very short paper. Someone literally writing a letter to the Lancet going, "Hey, this is something that you'd find interesting," and it is something we find interesting. Again,
It
it
doesn't seem to happen in all the patients,
no,
right? Yeah, because like I think that I love this figure actually. I think it's a good
yeah,
one. Yeah, like the red is the throat swab positive
yep,
and the yellow is the feces sample positive. So
Yeah,
not in all patients, but in a bunch of patients, they have this really long fecal sample positive range that happens after they become throat swab negative.
yeah. That moves us to the next. Uh, so intestinal permeability changes with bacterial translocation as key events modulating systemic host immune response to SARS-CoV-2. Colon a working hypothesis.
Right. So I think lots of papers were published right in this time,
Hmm.
taking these observations and saying, well, from what we know, right, like that model of the gut liver, right, or um, the gut lung axis, right, like we know immune is is linked. And
Yeah.
then I think also knowing that things can leak through the gut as well and they
Hmm.
can become systemic, right? If there is a type of infection or if there is like inflammation, right, you can get this like limited leaking through. Um, and yeah, and so the authors here they sort of start to build a hypothesis that and it's really linked again to just the same things we were saying. There's some sort of systemic response that's being triggered by that could be triggered by an intestinal infection,
Hmm.
so that there might be crosstalk between if you're carrying it in your lungs and you're carrying your gut. And I think maybe another thing you said that these places are linked, right?
Right.
That we would assume that if SARS-CoV-2 is able to infect the gut, right, which we see some preliminary evidence right now for that, it would happen like when you cough, right? Because then you cough and it's coming out, but then that's going by your trachea, right? So then you swallow some spittle, right, and then it
Yeah.
goes into
I mean, we're constantly swallowing saliva all the time, and sometimes, like when people sick, when people are producing lots and lots of saliva or spittle and stuff, they swallow a lot. So one, this is why I know this happens to me when I have a cold. Is one of the side effects is I fart a lot because I'm just swallowing lots of spittle and lots of air at the same time. So
uh-huh.
that's one way that the
Right.
bacteria just
And
bring
then
infection.
from that physical idea of like how does the virus get there, right? Then we can say, oh, and then there's also immunological crosstalk here as well,
Yeah.
because then it's going to be in a different zone. Your immune system's going to activate in a different way, right? And yeah, that activation might lead to a different outcome of disease.
Yeah. Uh, and it's interesting here because it also talks about bacterial translocation, which it also implies that there's some crosstalk between the microbiome in the system as well. Uh,
Hmm.
and
And
like,
I think this is sort of well, like, because inflammation is a non-specific sort of event, right? Like if you get a lot of replication that triggers the immune system to act in a certain way, it's going to let more of the immune cells into that zone, but by that action, it may also allow a little bit of the contents on the outside to get on the inside. So they're linked, and there's a pretty wide literature to say that that sort of leaky nature causes the immune system has evolved in that circumstance, right? To understand the signals coming from increased permeability over the epithelial barrier. Like epithelial
yeah,
should be keeping things out, but for many different reasons, it will change in permeability to allow like our cells to infiltrate outwards. Um,
yeah. And so this paper also uses it as an idea to maybe we should also target therapy to this. This proposes obviously the idea that this increased permeability causes the extra kind of symptoms with extra severity in SARS-CoV-2, I believe. Like the idea that it can so this is very much a review article, but it's proposing an idea based on the uh, data that was available at the time. Um, so in that way, it's quite interesting.
Absolutely. And then so then I pulled these two examples of just like
Oh,
we
Hamlin,
don't
I have them somewhere. But yeah,
I
I
pulled these two examples of just other pathogens that do infect the gut and then maybe to the respiratory or vice versa. And so one example is
cryptos.
cryptosporidiosis. And I'm not sure if people are familiar with this, but this was considered like one of the big killers in AIDS. because like you got immunocompromised, and then if you got a few of these, it's a parasite, I guess. It's a eukaryotic pathogen, but yeah, if you got a few of these spores into your lungs, it could grow there. But it's considered, first and foremost, it's considered a GI pathogen.
Yes.
And it's only in sort of some circumstances they are saying, oh, actually, we can find that it does. So, like, normally you'd control it, I think, as well in the GI tract, but because they
Yeah.
are fundamentally linked, this is what I was saying with the sort of like the coughing. Well, I guess this would be fecal oral, right? This would be like being not 100%, like it's very, it happens, right? Like
Yes.
there's spillover. And so those spores could get into the lungs, and then they could infect in that area. And this paper sort of reviews a little bit of the history also, where they're saying, could this also mean that there are strains that are more adapted for living in the lungs versus the gut? So I only pulled this as an example just to give people a sense that like this mechanism isn't crazy, right? Like to say that
Mm.
there's flow between the gut and the lungs, or the lungs and the gut. There are many ways it happens, and biology will find its niche, right? If it has the tools to kind of play around in there.
Yeah, and the second one is Yersinia pestis, which is the bubonic plague.
Yeah. And so this one actually, this is there is original research in this particular paper, because it actually looks at there are specific genes that are acquired that allow
Mm.
that switch to happen a little bit better. So they're looking at like a plasmid gene, and they say, oh look, like if you look at really old ancestral strains of if you look at old strains of Yersinia pestis, they don't have this plasmid. And when we infect mouse models with it, the type of disease it makes is a little bit different. So they take that as they try to build the story to suggest that the acquisition of this plasmid allows it to be a more productive lung infection than from a gut infection. And so, anyways, I think that like our reader seemed preoccupied with the idea, right, of what does it mean to have the gut as a reservoir. And I just want to point out that there are many stories, but it's like super complicated actually, like to uh, learn that causality. And you kind of also only learn of it in the past. There's not really like a good framework for picking up data to say like this is what carrying something in the gut means. Like this is what will be the answer. Like we have to look out for this particular thing, right, for it to transit between those two zones.
Right. And that brings us to intestinal host response to SARS-CoV-2 infection and COVID-19 outcomes in patients with gastrointestinal symptoms. So this is from one of the same people who was on that original evolution of antibody immunity to SARS-CoV-2 paper. So if you remember those pictures of the gut that were taken at 92 days were taken by the lab of Shorab Mahendru, who published this paper recently, which kind of
Mm-hmm.
follows up on that story about looking at what's happening in the intestinal tract during COVID-19. Um, so this is quite an interesting paper because, firstly, I was interested because the moment I didn't really, because it's very hard to pick out who does what. But when watching those HHMI talks, they did point out, okay, this is a person who got the data, so I knew exactly who to look for to see. Okay, let's see who's going to be expanding on this story.
Mm-hmm.
So let me pull up my notes for this quickly.
Replicating, I see that in figure. If you want to scroll down to a figure, so people can
Yep,
stare at them for a while. I
yeah,
think
yeah,
figure two, they're replicating the observations, right? That they can see SARS-CoV-2 nucleocapsid inside of EpCAM positive enterocytes.
yeah. See whether I've got the right figure or not, or maybe I've skipped it. Uh, figure five. Oops. Okay, right. I skipped it. Uh, figure. Oh yeah, there it is. Okay.
Oh, maybe this is where you saw the T. This looks like TEM.
Yeah, this is where I saw the TEM. Right.
Yeah,
Yeah, I got confused earlier. Because it was part of the talk, but this shows TEM of putative SARS-CoV-2 in the gut. But
yeah.
again, the gut has lots of microbes, so they say putative because they want to be like kind of have that asterisk over it. just in case.
Oh, sure. Yeah, like without a marker, it's hard to tell what things are just by their shape. There's a lot of different corona-looking viruses. I mean, that's the definition. There are a lot of coronaviruses out there.
So yeah, these are in human intestinal biopsy tissues from patients with COVID-19, and they
Hmm.
were basically taken about 30 days after the last positive nasal swab. Um, so these are in patients who have had the disease resolved. Um, and it has a microscopy with a presumptive virulent in there.
Ooh,
And
but I was just gonna say they do like a correlative approach in that TEM, I think. Like
right.
I think those uh yeah, I think those
Oh wait, yeah, you're right. I think those
those
might be
things
from
are
this.
yeah, I think they're from the slice that's like beside them, something like that.
In that case, they probably are SARS-CoV-2.
Yeah,
Um,
I think they feel more confident. They're still like again like I think this is they don't want to overstate what they're seeing,
yeah,
but that it's better than just doing a slice out of nowhere, right? And saying I see corona-looking viruses in that area.
yeah. But I mean, the bizarre part is that they tried to. Oh, God, let me see whether they wrote this, because yeah, no infectious variants identified in gastrointestinal tissues of patients with COVID-19,
Mm,
which seems to contrast the image we just saw previously. Uh,
right, it's like virions, like whole virions.
so we saw whole virions, but apparently those virions don't seem to be very good at actually causing infection. So they saw them there, but when they tried to put them on the plate, those virions just sat there and didn't cause an infection, which
Yeah.
again is very odd behavior. Um,
Mhm.
uh, yeah, and what they also found was that I think that the severity of the infection was reduced as well.
Mm.
Uh, so in patients who had these gastrointestinal kind of symptoms, the overall thing they found was that the severity was significantly reduced, uh, which I'm
Yeah, that story is not so crazy actually. I feel like from and that's why it's nice to go back to that review, right, that says there's crosstalk between these two areas, right, through the blood. And if we think about the gut as somewhere that actually has to be more uh, less overreaction, right,
mm,
to outside things because it sees so much foreign stuff. There's like the microbiota that grows there is like wild
yeah,
inside the gut. Lots of things that are always rushing back past that epithelial surface. I think people would say that the gut produces more tolerant immune responses than in other locations because it can't live at such a high level of inflammation all the time if it's going to inflame every time
yeah,
something goes by. And so a hypothesis, right, that you could draw, and I think that this paper is trying to advance, would be that that type of immune response happening in the gut means that you will also less likely to have a very severe immune response when in the lung version of the infection.
yeah. I can get that. I mean that explains that does make sense, and it does also counteract that review that we talked about, I think not 5 minutes ago, about the increased gut permeability making gut infections more susceptible to severe disease.
Mhm.
So it is interesting to see this contrast that
Yeah,
review with
because I
actual
think
data.
yeah, like that the hypothesis before the increased permeability, like yeah, they were thinking those pieces would then get systemic, and that's going to cause this systemic inflammation.
Mm.
Yeah, but maybe that's not the case, and it's actually protective. So working with what data we have, right? You can't say right now. Like someone has to go in and do the exact experiments that answer these types of questions to give us a sense of certainty. And so right now, seeing data from the hypothesis where it's lessening, right? I think that that's helping boost this sort of
Yeah,
interpretation, but it's still not. Yeah, we still don't really. I don't think anything has definitively given us the end of this story yet.
yeah. I mean, there's a lot of like interesting bits and pieces in there, but I think the dreaded more research is needed is ever present.
Yeah.
It's
Did
like,
we
why is
did
more
we
research needed? Weren't you doing your job? I'm like, well, job never ends. We're scientists. We're
Well, that's true. More data only really brings more questions.
yes.
I think we may have skipped a few papers. Did we? We want to go to SARS-CoV-2 productively infects human gut enterocytes.
Yes, we do. Yes, got it. Yeah.
And so it, people have followed up on those that very early, right? We had that you had that March paper
Hmm.
where they had biopsies, and another thing that could help us confirm infection is just to infect things that we think should be infected by SARS-CoV-2 and see what happens. So in this paper, they use organoids
Mm.
actually, which we haven't really gotten to talk about too much, but they're a step up from cell culture. They're a bit more differentiated. So we think of them as being more similar to cells that we would find in vivo. Um, so first, yeah, so they do it on airways, which we do know,
Hmm.
right? That airway gets infected. We have all that lung pathology to tell us. And then they move on to organoids, and they show that those organoids also get infected. Um, so they make these balls of intestinal cells and they place SARS-CoV-2 over them, and then they see, oh, they're
Is
picking
this?
up SARS-CoV-2.
Yeah. And yeah, we've got even some electron microscopy of the SARS-CoV-2 infected
Mhm.
organoids. So it's
And in this case, we know it is definitely
we
SARS-CoV-2
know.
because these organoids never got like they're not in a natural environment. They're in the lab, right? We're putting SARS-CoV-2 on them, and so like the criticism of this type, right? Like to get at these science questions in science, you come from different angles, right? You can
Hmm.
try to reconstitute the system to see what's going on, or you can try to break down the system and find the needle and yeah, find the needle in the haystack essentially.
Yep.
And so we're seeing here the other approach, right, to get at this information. Let's reconstitute and see if it's possible even for SARS-CoV-2 to get into these cells. And yeah, so they're showing that it is here.
Yep. And that brings potential intestinal infection and fecal oral transmission of SARS-CoV-2.
Hmm. So I think this is it's actually a review of us or like a sort of it zooms
Yep.
out. They do a really good job of pulling together tons of studies that report gastrointestinal symptoms. Um, I think it just uh, investigating this little side story, right, about is there gut replication just shows us that so much research was coming out. Everyone is trying to keep up, and even scientists are publishing for themselves, right? These these summaries of
Yeah.
everything that was done. So it's well known that there are. What I got from reading this is that there are lots of reports of gastrointestinal symptoms. So if you combine that with the understanding that it is possible for SARS-CoV-2 to infect the gut, I think that it brings a very strong I think it brings a strong suggestion that there is this infection, and yeah, we don't know. And then, as you were saying in the paper that like two papers that we just read ago, like that that gut infection though might impact the lung infection.
Hmm.
So you might have gut symptoms, but then your lung symptoms are going to be modified by this particular format of
Right.
infection.
That's now in let's see SARS. Oh, hold on a minute.
Oh,
SARS-CoV
sorry. Yeah.
two. Sorry, SARS-CoV-2 induces a more robust innate immune response and replicates less efficiency than original recipe SARS-CoV in human intestines. An ex vivo study with implications for the pathogenesis of COVID-19.
Yeah.
So,
So I think I brought this up again just as, we want to have multiple angles in order
mm,
to say that SARS-CoV-2 is replicating in the gut. So we've seen observations from biopsies, which are really strong, I think, sort of samples. We've seen we've seen organoids, right, a sort of reconstituted cell culture system. And then here we're seeing it in ex vivo. Uh,
yeah,
so that means that they did the biopsy, but then they're growing the biopsy in media, and then they're infecting it in the lab.
yeah, and again, this kind of finds that SARS-CoV-2 can infect these cell types, but not as well as the original version of SARS-CoV, which is, I mean, it's an interesting comparison. Uh,
Yeah, but I think what's useful then about that thought is that it's bringing in this thought about um, this family of
mm,
viruses, right, has some propensity to infect the gut epithelium, and so it's. I think again, I think our listener was preoccupied or like focused on this idea of what does it mean as a reservoir for SARS-CoV-2 that it replicates in the gut, or yeah, it seems like there's this evidence that there's gut replication. Well, uh, I think this is part of that story. I
yeah,
don't know exactly how I would start spinning those threads, right, and like saying what paper I'd like to see connecting those dots, but the fact that there are the ancestral strain, right, of this virus, or rather, I guess not ancestral. It's like a cousin, right? It's a
yeah,
it's a branch that came off at a different time. We just saw it earlier in the course of our history, but from the same sort of pool of the family of viruses, the ones that make the jumps into humans, right, have this relationship with our gut tissues, and yeah, that speaks to the I guess the potential right inside of the family to have productive infections and also to switch between hosts maybe.
yeah. And I guess this might be interesting from looking at from the zoonotic perspective of the actual animals that these evolved in. Because I think I remember hearing early on about the how like bat feces it was SARS-CoV-2 was carried in bat feces originally, or and that's I mean, again, this is something I know very little about, so I am speculating into the void. But the thing is, sometimes you get certain viruses that are adapted to like a gut immune system that suddenly are now able to infect the human lungs because they're very similar
Yes.
areas. So there's
Yes.
um,
Yeah, and I wish I pulled maybe now I wish I pulled another example, but I think that that's something that's really well known in the flu field,
yeah,
right? Because we have these examples of flu in birds and pigs, and in birds and pigs it is, or in birds at least, I know it's primarily a gastrointestinal
yeah,
disease in the birds, and then when it transitions into respiratory, that's just because the birds' guts are kind of have some of the molecular signatures of our respiratory system, but like the type of evolution that happens, right? It still means that birds end up being an evolutionary hotbed, right? Yeah, they end up being a hotbed because they have the capacity to support this replication. Things get reconfigured, and then when it makes the jump into humans, that's when we get this. Is it more or less severe based on some of these markers on the outside?
yeah,
so, so I sorry, I just want to again in the speculation realm, right, looking at some of this data, we could say, oh, maybe right, low level human gut replication is where we're going to hold the season out. Like if SARS-CoV-2 ends up becoming endemic, the path for endemic could be through this mechanism, could be through lower immune, like sort of less severe immune responses based on both vaccines and getting it in your gut, and then you never know when that might pop up again as like a seasonal strain that comes out. I don't know,
mm,
that's just like, again, that's wild speculation, but these are the types of, I think, thoughts that one can have. I think the real challenge for scientists would be what sort of experiments can be done in the bench to inform surveillance.
yeah.
uh, right, so like if we do see variants coming out, can we link variants to this somehow? I think that's sort of like the place that would be interesting to look at.
Yeah, indeed. So
Okay, through this.
l-
I
so
think we're going to focus a bit on the immunity side. Yeah.
Yeah,
Okay.
we're going to be focusing on immunity side because I think this also comes from one of the co-authors of the original immunity paper, and this one where they focus in on the T cells. So I remember when we were first talking about the paper, they were focusing on B memory cells, but this one looks at the T cell memory response as well, and this also looks at the T cell responses uh, at 6 months after infection, and looking at and does some of that like high dimensional data analysis that we're so fond of here, where
Yeah.
they take a look at these blood
Just
samples
like that we ogle.
that we yeah they look like they're really beautiful pictures. I I need to like take a good like week long run up to figure out what they mean though, because yeah, they each of these clusters relate to a specific cell type, and then this kind of shows what happens, how those cell types broadly change over those months. Again, it looks beautiful. No idea what it means yet. You need to really delve into these papers and immerse yourself in them before you can really figure that out. So since we're doing this like quick skimming, I think mostly what they look at is the shifts in the. Sorry, did you say?
I'm good.
Oh, sorry. Okay. So mostly we're seeing like shifts in the T cell compartments in terms of memory and how long they persist for after SARS-CoV-2 infection. Um,
Hmm. And once
so
again, it's like they see long term. This like they have a 6.1 months post
yeah.
infection time point.
So yeah, they again see long term, and I think this links into the idea that this response is being maintained somewhat. Uh, so uh, so I find this paper quite interesting because it it does go into that T cell side of the story that I feel is often kind of neglected a bit. But also,
So this paper would, oh, go
sorry,
ahead.
no, you go for it.
Oh, I was gonna say, so this paper would is sort of is extending what we've talked about a lot on the B cell side, or the observation that we talked about earlier that there's this there's a change in the neutralizing ability of the of the serum of people uh, over time. This
Yeah.
links it also to T cell responses, right? That they're also changing, which
Yes.
I guess like should be like yeah, that should be understandable. Do you know if they looked at I can't find it. Do they look at like the specificity of these T cells to SARS-CoV-2 antigen?
Yes, they do. So
Or
these are, I think these are pretty much a lot of these are T cells that react to SARS-CoV-2, and then they look at the various markers that they express, including exhaustion markers, which is an interesting thing because that's another part of the B cell story that we've been delving in a fair bit deeper into. I think we covered it, I think maybe a month ago, where we looked at exhausted B cells and how that
yeah.
is affected by vaccination. Um,
Oh yeah, I see. Yeah, they use different peptide pools from a whole bunch of different regions of the spike protein, it seems like, in figure
yeah,
five.
Figure
Yeah.
five. Let's take a look at Figure five.
Yeah, so this is very similar to the sort of stuff. I mean, similar like in the zoomed out sort of view,
Mm.
right? That there's some difference. There's a difference in the way that memory cells are responding to spike antigen or SARS-CoV-2 antigens as the infection progresses. But not even that, like sort of even after the infection progresses.
Yeah, uh, totally. Well, so the next paper was looking at again immunological memory to SARS-CoV-2 assessed for up to 8 months after infection. Now, I posted this paper because this is very because they this looks at a bunch of samples from people. So they took 51 subjects and got longitudinal blood samples to look at how the SARS-CoV-2 specific immune memory develops. And the interesting
Mhm.
thing that they see here that we've also seen in previous papers is that the memory B cells against SARS-CoV-2 spike actually increased during the
Yeah.
between 1 month and 8 months, which is what we've seen previously in that paper we covered a few on like pathogenic B cells, I believe, and also
Do you remember if they looked at that far of a time point out though? I thought that this time point is this is the paper actually that says the B cell observation. So our listener, to me, the listener pointed out both this paper and the intestinal staining paper as building this thought of changing or of the reservoir in the gut, uh, and and so to me this is the mirror to the T cell one.
yeah.
But I don't remember if in previous T cell papers they looked as far as 8 months out.
So I remember in the B cell paper we looked at, they did look pretty far. I can't remember whether it was as far as 8 months, but they definitely because in that paper they waited for, so it was after the initial infection. Then they looked at people who were just about to get the vaccine, and then
Oh yeah.
so
Hmm.
I think it might have been around that time. I know it was a very long time, and it was surprising to see a spike in B cells. And also in that paper they found that a lot of those compartment was taken up by exhausted B cells or pathogenic B cells as well.
Yeah.
And I think
No.
that. So
for the best antibodies, right? They're just making good enough antibodies.
yeah, I mean that was our idea of the paper when we read it, that they were just creating these. But another thought could be, if we look at through the lens of this, what we've been talking about today, is the idea that maybe there is a continuous SARS-CoV-2 process that is causing this response to continue ongoing.
Hmm. Hmm.
So it's not just that the pathogenic memory B cells are kind of propagating themselves, but they are being driven somewhat by the continued presence of SARS-CoV-2, even if it is in this kind of dormant state apparently.
Hmm.
Um, so that, I mean, that this is kind of interesting to build on that. It allows me to talk about that story without revisiting our previous paper too much. Um,
Yeah.
but yeah, I mean, they found quite a lot of people were positive for CD4 T cells as well in this paper as well. It declined slightly, but immunity is quite complicated. So this is where my memory my brain's melting a little bit at this point. um,
Hmm.
next slide is
Yeah.
ah.
Move on.
Now this was quite an interesting one because I think I'd have probably wanted to talk about this even if we weren't talking about this entire thing, because this is called a prior SARS-CoV-2 infection rescues B and T cell responses to variants after the first vaccine dose. So these are people who are,
Interesting. Yeah.
yeah. So they looked at people who were infected with the original like Wuhan strain of SARS-CoV-2 and looked at their B cells, and then they also looked at kind of how those changed after the first vaccine dose. At least that's if I'm interpreting this paper. But I think they might have just looked at it after the first dose of vaccination, and
Hmm.
then they tested it against new strains like the Kent strain or and the South African strain, where and seeing how well they neutralize it. And they found that people who had that prior infection were more likely to neutralize these new variants compared to people who had just had the vaccine, um, which which
That's very strange. Because I remember we were saying for neutralizing wild type, that's not, or the titers are higher,
mm,
right? For if you were
vaccinated.
if you were, yeah,
Yeah,
vaccinated.
that is what we've been looking at, and so this
Hmm.
is like very interesting because it poses a question: what matters more to the immune response? Is it the quantity or is it the quality?
Yeah.
And
Mm.
so the implication here seems to be that the quality of antibodies you get after a SARS-CoV-2 infection are enough are different enough that they kind of can grab these
Well,
variants.
like no, it modifies the quality, right?
Yeah.
Uh, it sounds that when you get when you get the vaccine and if you've been previously infected, the quality of the neutralizing antibodies that come out after that is different.
Yeah. So this is interesting. I mean, I haven't delved into this like enough to get a concrete answer on what this paper's saying about that, but I I believe it'd be quite an interesting one to look at from looking at the different ways we can interpret that data.
Okay,
Um,
so yeah, I feel like this is a good cap and to end the. I guess we've spent like almost an hour kind of plotting through these papers, but um,
yeah,
at the end of the day, what do we? The thing that we missed from originally reading that evolution paper was the observation that uh, there is some replication, or there could be replication in the gut. And since then, or like even just looking back into the history of the literature now, we can see that there's pretty good evidence that there is some sort of gut replication, and um, that it correlates, or we have one study that correlates that to better responses, right, less severe. We know that probably correlates with the fact that you shed a lot afterwards. If
mm,
shedding RNA in the fecal samples kind of correlates to this phenomena. um, we have a sort of a framework for understanding that the immune systems are different in those two zones, but they have crosstalk through the blood, and so that would lead to some sort of thing. We've also kind of spoken about a little bit about evolution, right? That like uh, you could get adapted to the gut maybe, and you could only grow in that scenario. But that could end up being a way of being a reservoir for ourselves. But the experiments to prove that sort of stuff are kind of difficult to find out. Um, or I can't. I can't imagine what I would want to see to prove to me that. So I think we gotta wait for the field to give us some more insight. Is there anything else that we could like do to summarize that sort of
Well, I
what we?
mean, I'll throw a curveball at you, Danny. Should this make me more afraid that if we have long-term gut bacteria, I mean, is this something that should should make me
No, I mean
worry?
to me, this is pointing out in other places. This is just part of nature, right? Like
Hmm.
we have stuff that grows all over the place. It seems like our and at the end of the day, that's not. We can't plan for that part. Like we can just use the information that we have and move forward,
Hmm.
right? So the vaccines work to remove. I guess we don't know how the vaccines even impact this particular thing, right? How does being vaccinated impact growth in this region?
I mean,
Uh,
well, we only really have one paper that seems to be showing it at like at that long time point. I think maybe two papers, I'd say.
mhm.
And I think there is a lot more work that needs to be done on this. But from basically, this is interesting from like a basic science research perspective. But we haven't seen anything in the clinic that seems to be indicating this is an issue. And I think that's the important part. That
I think it's more this, yeah, it's definitely this is very basic. I think it informs surveillance
mm,
strategy, right? Like, I think it might be saying that maybe we should surveil the genomic variants that come out from fecal samples, right? Maybe fecal surveillance might be something useful to do. Um, but yeah, there's no story emerging at least of what will be the like is the stuff that's coming out of our gut like just going to give us the same high level of infection, right? Like will it produce all these outbreaks? We can't say that.
I mean,
And it doesn't.
at the moment there hasn't been any like evidence of foodborne transmission like in real terms, but even theoretically
Mhm.
it's possible. But it's weird if it but it's weird because like I mean I guess that from the Meharandu paper where they found that it's producing these variants, but those variants are non-viable, and that's
Mhm.
an interesting finding because because viruses can go dormant and go get silenced during infection.
I
Um,
mean, they could be adapted for a different scenario, right?
yeah,
I guess I don't know how they exactly did their plate test, and this is where the real devil in the details comes out. Where like, how did they do the plate test where they showed that that wasn't replicating there? Because maybe they still grow in enterocytes, so that's just its new environment is living there.
yeah,
Or maybe it is just that it's like a dead end for them. They grow there and they kind of sit around, but nothing more comes of it. Um,
yeah.
I mean, there are viruses that hang out for a long term. I mean, those are things usually that get closer to the genome, but uh, yeah, that's also a possibility.
So yeah, I think the thing we should tie up with this is very interesting from a research perspective. From a public health perspective, it just doesn't seem to be something we should be worried too much about.
Hmm.
Um, but it's from as researchers, we're very interested in finding out like where this last kind of bunker of SARS-CoV-2 that lingers afterwards, what happens to it, and what, why does
But
it linger
but
on?
I
And
think it does. No, but I think it the only to me the interplay with public health is surveillance.
yes,
Like everything on the survey like and that's true even for the variants,
mm,
right? Like I think that the interest of the variants, all thing we talk about when we say we're interested in variants, it all just goes back to surveillance because we know the tools that we have to suppress serious infection and suppress outbreaks, right? We have public health initiatives like distancing, masks, and distancing, masks, and lockdowns. We have a vaccine that reduces hospitalization. So those tools are
mm,
very much in place, sort of to deal with the negative outcomes that we feel are coming from the virus. And but but in terms of trying to get ahead of like
mm,
something potentially on the horizon, all this research would flow into that. So stuff about variants, more transmissible, more this, it flows into modifying some of those procedures, maybe figuring out what should we be looking for in different localities. How do you modify your procedures on those localities? Yeah, and this is just another part of nature. I think to me, when thinking about SARS-CoV-2, I do feel that it is becoming an endemic virus based on the response to it, and it may also be because of the stance. Actually, maybe this is a really good segue into the next paper, unless you have something else
yeah,
to say.
go for it.
Okay, that's a really good segment in the next paper where we have SARS-CoV-2 elimination, not mitigation, produces the best outcomes for health, the economy, and civil liberties. So this is not like an in-depth research at all. It's just a opinion piece,
Mm.
basically, where they did a bit of modeling as well to look at, or they did some data collection to look at how GDP relates to um, SARS-CoV-2 outbreaks, and they categorized countries into uh, two pools, ones that are focused on eliminating the virus. So the policies are all designed to get virus down to zero versus policies that are just intended to make sure that not too many people die from the virus. Um, and they actually, in their data or in their small observations, they show that it's better the policies that have come out, or like, yeah, for the countries that have adopted policies that are focused on elimination, they do better in the metrics of how many people actually end up dying, how many people, or what's the GDP of right those countries, or I guess it's comparable on the GDP side, and yeah, it's like yeah. So all that to say, uh, I think that this information about gut replication, right, sort of feeds into this sort of stuff. Like it all feeds into policy at the end of the day. And for the daily person, right, I don't think that there's a lot you can do to control some of these things, especially since it's in a sort of like an unknown, in a very unknown space. Um, yeah, that's all I have to say about that. Do you, how's
Yeah,
it sound to you?
I don't really have anything to add to this, but I mean, this is an interesting for because the hope is that say someone who has a role in developing policy reads this paper or they pay attention to this because I think that's maybe the main target audience for that, and also to kind of help build up that discourse because there because I guess when it comes to economic mitigation, people are very short-sighted in politics, where they decide to do things that are good for the moment, but actually, if you look at things over a long term, so mitigation in the short term, it means that we save money on things, but actually, the long-term outcome of having SARS-CoV-2 be a constant threat for the
Yeah,
next 5 years or so is much worse than if you take a much bigger hit earlier on and limit it.
right.
And so that's
And that's why I wanted to segue into this from talking about this reservoir stuff, right?
yeah.
Because I think that the policies, because there is this unknown element to the biology of SARS-CoV-2 that we're still uncovering, it means that taking a mitigation strategy may not, it, it might be that just by chance, right, that the stricter elimination strategy is somehow limiting some of the biological possibilities of the virus in a way that is better for outcomes long term on the health secure system.
Yeah. And
Yeah.
okay, so speaking about like mitigation strategies, our next paper is about manure.
Yeah. So, anyways, yeah, that's. I guess we spoke for a whole hour on this interesting thing. I guess we should say thanks to the listener who
Yeah,
brought
thank you,
that content.
Minimal. They've got a YouTube, so they piped up in the comments in I think uh, a couple of videos ago, but they've got a YouTube channel themselves where they look at some. They do some cool stuff.
Yeah, excerpts from HHMI talks. At least that's the stuff that I saw in it. Yeah, I think it's great to have other people reading along with us, and it gives us stuff to look into as well, and give us a sense of what's going on. We hope
Yeah,
that our interpretation is helpful for others.
and I
And
mean, we are just skimming these papers, so at the moment we don't have time to do a deep dive on each one. So there is definitely going to be stuff that we miss. So having comments like that pipe up to point out interesting stuff that we miss can be so useful to us.
yeah, absolutely, absolutely. But plowing through, we also want to cover some interesting papers that we might want to cover non-SARS-CoV-2 related, in case that that is more resonant for us or others. And the first one that I picked out that we're going to talk is manure microbial communities and resistance profiles reconfigure after transition to manure pits and differ from those in fertilized field soil. And so I'm sure people are familiar with the idea that we fertilize our crops with manure. You may also be familiar with this idea that uh, there's antibiotics used in livestock and
Mm.
that makes there's then selection then for antibiotic resistant bacteria inside of the guts of these organisms, of these animals. And so there's this method of, I guess, mashing all of the manure together and doing a sort of crude fermentation in a pit
Mm.
before you take that manure and you spread it over the fields. And so these authors look at they look at those the prevalence of antimicrobial resistance genes in those pits, and then also how that interacts with the diversity in the fields that they end up being spread on. And they find that actually there's no modification inside of those pits, but the field microbiome is pretty resilient to not getting overtaken by those specific antimicrobial strains, resistant strains. So I don't know. I feel like we still shouldn't really be giving antibiotics to our animals. I think there should be a
Yeah.
push against that. It doesn't tell us that it's all fine, right? That we're not creating more antibiotic resistance through this practice, but it is saying that it's sort of, I guess it's saying it's complicated.
Yeah.
Even though we're making that antibiotic resistance in these pits and it's not really getting degraded at all, when you look at what's going on in the field, it's not like changing the microbiota of the field, but I guess it is bringing those genes out there for horizontal transmission.
Yeah. I mean, it'd be interesting to see like what kind of effect this antibiotic resistance has had on the wild species that use those antibiotics to fight off their competitors. Because I can't
Yes,
imagine it's
yes. I think that I think is maybe what they're speaking to more, that like it doesn't change that scenario out there.
yeah, which is
Yeah.
interesting. Um so yeah, what else we got yeah, so based on that we've also come for a paper called Reconstruction of Ancient Microbial Genomes from the Human Gut. Uh
Yes. Actually, you can see that like certain things also just like we were talking about poop, so it's like, oh, manure paper. We were talking about gut stuff. We were talking about when I was looking at that Yersinia paper, I was like, oh yeah, like people do this. They look back at ancient samples
yeah.
and they try to understand what's going on. So this is one of these this like sequencing old stuff.
Yeah. Paleofeces. It's not something because museums collect a lot of different things, but I didn't really. But I only now like paleofeces are an important factor in that because finding out how people lived, what they ate, is very interesting. And so
Absolutely.
yeah,
And I don't know if people have heard of things like, we have like Neanderthal genomes and stuff. You get that from like preserved matter, right? So there's all of these techniques now to extract DNA from what you would think is unextractable
yeah,
rock-like things. And so yeah, they do that here, and yeah, they find some stuff about the genomes of the past. I think there'll be a lot of technique also in this paper, like just to understand what are the challenges of reconstructing a genome from the past. But ultimately, I think they're interested in seeing like, uh, I guess they look at industrial versus non-industrial. So
yeah,
they try to look at
because you want
how
yeah.
shifts happen. Mhm.
Because yeah, I guess you want to compare like with like, and so you're imagining that these people in the ancient like times didn't have Mars bars or other sorts of candy, but they did have a lifestyle very similar to other people who are living in similar, and so that'd be the ones to compare with. And I
Yeah.
think they find some bacteria that aren't found in various microbiomes. So I mean, there is this idea that microbiomes are passed between like mother to child,
Mhm.
and that there's some kind of conservation there. But another idea is that maybe they're interacting with their environment, and I think this paper kind of indicates that environment has a big role in a bigger role in the microbiome than we might have originally thought.
Yeah. I would also say that part of understanding that is just the understanding of how dynamic it is through
Mm,
time, right? Because there is a sense that it's dynamic in us in our time scale, but what about in the larger time scale, right, of history, where there's yeah, a lot more shifts going on. But I think that at the end of the day, these are probably like incomplete. They said that yeah, they're trying to reconstruct whole genomes, right? With the idea
mm,
that um, you can't just do this by identity to say like, oh, certain microbes are missing from the past, because the function of those microbiomes is what we also want to know. Like, how does the function change over time, if at all?
right.
So this sort of, yeah, whole reconstruction is an important an important way to look at the past, not just cataloging what was in our guts back then.
Yeah. Uh, so now that let's see, what did I put next? So next we looked at the Coxiella burnetii QPH1 plasmid is a virulence factor for colonizing bone marrow derived murine macrophages.
hmm So this actually, I was thinking about like unculturable things, and
Mm,
when I was in grad school, there was a lab, and it had just recently found that you could culture Coxiella
mm,
because it's an intracellular pathogen, so it lives inside of cells, and that's like not easy to do, like to grow
mm.
cells and then grow your bacteria of interest inside of those cells. So what had recently been found was a way to grow it just with like regular medium. And since then, there's been lots of research around it. So here they're talking about a virulence plasmid. I think it's cool too. It's talking about that evolution of virulence, right? You can have
Yeah,
these genes that uh, go on plasmids, and when they move into a strain, they give that strain a new ability, a new environment that it's allowed to grow in. So yeah,
yeah.
that's why one
I mean, this
kind
is
of.
interest. Yeah, this is another big step for the field because Coxiella has these plasmids that are either like replicating or in the chromosome, but they found a way to pull those out, and then they can transfer them between different strains, which means they can actually figure out how the virulence works, which is quite.
Yep.
So this is probably going to be quite important for the that field for a while, I think, because lots of people
Yeah,
will be
it's true.
using these
It gives a
tools.
lot of meat, a lot of genetic meat to start to disentangle.
Yeah. Uh, moving on, because I think we've got so next one is going to be intracellular antibody targeting. I think is the uh,
Okay.
okay. Let me see if I can find it because
Oh,
I might have. Uh,
okay. Because
oh
I
yes,
was
got it.
okay.
Sorry. Yeah, I was panicking for a moment because I've got a million tabs open. I'm selecting between them. So yeah, an intracellular nanobody targeting type 4 secretion system effector inhibits Ehrlichia infection. So again, we're looking at another intracellular pathogen. This time it's of a tick-borne disease that
Mm.
infects macrophages and kind of obligately lives inside of them. And this stood out to me because nanobodies. We've been talking about nanobodies. They're like antibodies, but much more compact. And
Mhm.
and this kind of uses that compactness to attack something that's inside of a cell, which is
Ah.
quite
Yeah, I didn't
interesting.
think about that. That's super strange. So it somehow
Yeah.
gets it gets uptaken, and then the type 4 secretion, oh, it's an effector. So it just
So
goes into
yeah,
the cytoplasm and neutralizes this thing that got secreted into the cell.
yeah. So Ehrlichia produces this type 4 secretion effector to manipulate the cell that's inside, and
hmm
this nanobody basically binds to that and stops it from happening. Um, But yeah, no, this really stood out to me because I'm like, how does an antibody get in? Apparently, they've got some clever. They link it to some clever chemicals that
Yeah,
effectively,
that is very interesting.
yeah, and they pull it out all the way into a mouse model, which is again a good demonstration of technology because, again, like antibodies themselves are quite finicky,
Mhm.
especially when you're talking about intracellular. So you've got like things to think about, like your dosage and actually how much of that dose actually enters into the cells. So it's very interesting from like a drug targeting perspective because people looking that these nanobodies for treating SARS-CoV-2 and various other diseases. So I feel like looking this might be able to tell us about how those other sort of treatments might go.
Absolutely.
And then now next is colistin. Colistin kills bacteria by targeting lipopolysaccharide in the cytoplasmic membrane. So
So
colistin,
this is one of the things where everyone knows that it's like building upon something. It's getting like a finer detail to things that we already know, it sounds
yeah,
like. hmm
yeah, it is. Basically at the moment it's established that that colistin disrupts the bacterial outer membrane by targeting lipopolysaccharide, but
hmm
they wanted to figure out like how well this interact how this caused bacteria to die. Um,
hmm
because sometimes you get antibiotics and we know that they kill bacteria, but we don't know how until much later. And this is
Yeah.
one of those sort of situations. And what they found is that also there's something happening on the cytoplasmic membrane as well. And and based on what they found, they kind of added another new antibiotic into the mix called uh, muripavidin, which causes lipopolysaccharide to build up at the inner membrane. And then when they
Oh,
kind of
as a
combine these,
synergistic effect then.
yeah, so they found some synergy between these two that
Mm.
made the bacteria just pop open much more easily.
And that's really common these days in the, I think when people think about antibiotics is like trying to use combinations of them that
Yeah,
target either like that have some sort of reason why they're together, right? I know that people do beta lactamase inhibitors with beta lactam antibiotics.
yeah,
So like this is kind of a similar thing. Together they're better. But they come to that understanding through understanding the mechanism, right?
yeah.
Yeah.
So
So
this is
that's
very much a mechanistic kind of paper, and I mean, I guess the interesting part is that synergy and how that understanding led to the kind of understanding of
mhm.
mixing these. Uh, and I guess mostly I saw it was an eLife paper and thought this will be an easy thing to
I was
read and
gonna say
review.
that's another, yeah.
Just
And
because I've
we've talked about a bunch of antibiotics, right? But
mm,
over the past few as well.
yeah,
Uh, so we've seen some of these assays. We're familiar with them. Okay, what else
yeah.
do we have? I think
I
did we skip structure and dynamics of? Oh yeah, you got
There
that one.
is, yep,
Great.
structure and dynamics of a mycobacterial type 7 secretion system. So,
So I think we type 7 secretion has come up a few times now in the news
mm,
articles that we've been bringing up. Secretion systems are super important in bacterial pathogenesis. I mean, we just said an effector of the type 4 secretion system is important in Ehrlichia infection. So there's a lot of interest to know what these things do. They're also huge complexes. And so using cryo-EM, I think this group figures out some structural features, figures out a structure for this type 7 secretion system.
yes, they do. So I mean, cryo-EM, it's just, they produce such brilliant images. So that's,
Yeah, brilliant images of a really giant like complex of proteins that's important for pathogenesis.
yeah,
And also like it's 7. I don't think there's an 8. Like this is the most recent secretion system to be described uh,
yeah.
for bacteria.
So, and I guess the final paper I was going to point out was something that we've talked about before, just a kind of
It
the
got published.
malaria vaccine. It's been
Yeah.
published now.
Hmm.
So,
Yeah, we previously talked about this in a news article. I think it was a preprint at the time.
yep,
Um, and yeah, it's nice to see that it's being published. Interesting because it's been very difficult to make a vaccine. I think I said in that art in that episode that like I want to know about the technology of it, but I don't necessarily want to read a clinical trial about about vaccine.
yeah. So I guess we're gonna have to just think about which of these papers we'd like to choose.
Yeah,
Um,
do you think we should do SARS-CoV-2 or non-SARS-CoV-2? I think that's one of the big
ooh,
because I there are some SARS-CoV-2 ones that would I think that like some of the I'm not sure, but uh like
so
one of the later SARS-CoV-2 ones might be interesting to look at.
yeah, so
Um,
there's the the prior SARS-CoV-2 infection maybe, or
yeah,
the
the one that tries to link the severity um to severity to the gut
oh,
infection.
actually, yeah, I'd be up for that. So that's the Randu paper. Let me see if I can. But yeah, no, I know exactly the one you mean. So that would be yeah, I'd be up for that one. Uh, because yeah, that's I've got
Paper.
I so yeah, I can't
We
I
like lost the
I've
things. I'm
l-
trying to look for. Do you remember what the name of it was? I should have kept it open. I like close my articles as we move through them.
yeah, hold up, let
Yeah.
me so this is the so our viewers must be thinking, oh, this is great. This watch two people read read a screen for a second. But you know, I believe it's called Intestinal Host Response to SARS-CoV-2 Infection and COVID-19
Oh,
Outcomes in Patients with Gastrointestinal Symptoms. Uh,
gotcha.
so, uh,
Yeah, so that's a possibility.
Yeah, I'd be up for that because I think it's got the right mix of like interesting things. It allows us to delve into the story a bit more,
Mm.
and we have put quite a lot of time like looking into the background of it. Um,
And I mean, just to fill out this, yeah, this particular interest, yeah, it does seem like it has a lot of the different pieces about the story altogether. Okay, and then yeah, from what else do we have?
so, uh, I mean, I think the Type 7 secretion system is a strong candidate from the like glorious clockwork that we're seeing here. Uh, I got addicted to another YouTube channel. Glorious Clockwork is amazing.
Yeah.
Uh,
Yeah, like just yeah, seeing molecular, like they call them molecular machines, right, for a reason, because they
yeah.
just have components to them.
Uh,
It might be like a really easy paper to cover because it's just like looking at pictures, a lot of discussion, right, about like what does it mean that they saw that this touches this, and
yeah.
people get meaning from structures.
Yeah. I mean, I guess the main challenge here is explaining the paper because there's a lot of like language that needs to be translated for this because you've got ESX5, ECB, ECCB5, things that I don't know yet. I'm gonna have to look into. Um,
Yeah, tons of protein names, right? Because all that
yeah,
that structure is made up of tons of proteins. They all have their own unique names and have been studied in their own labs and stuff like that.
yeah. So there's that kind of challenge, but yeah, it is very hyper focused on one small area. The intracellular nanobody, it's a treatment strategy and it's kind of interesting, but I don't think they
I
go.
am interested in that one, but uh, but again, like I'm so, it's like I want to find the ones that talk about like how does it get into the cell,
Mm,
right? Like how did those chemical things like work? I feel sometimes we never come across. We always see like the story in its later stages when they've like executed successfully like a therapy with this. It's still on the bench of this case, but um, yeah, I still just I want to know more about like the initial technologies always.
mm.
uh, and then there's also like past stuff. I mean, from our top 10, there are still things in the top 10 that we haven't done. Continuous evolution of Cas9 variants compatible with non-GPAMs. I thought that was a very interesting one.
Yeah, that's good from a bioengineering perspective, where they take
Yeah,
Cas proteins, which are these gene editing proteins, but they are limited somewhat, so they force them to mutate to have new
the gene-directed
functions,
evolution. Yeah.
basically. So that would be an interesting paper from the perspective of the bioengineering story.
Yeah.
Um,
But I have but I do I think I said this last time that I want to move to like something that's like more mechanistic.
right, yeah.
With actually, maybe intestinal host responses really is the mechanistic,
I think that
right? There
really is. Uh, Paul,
is immune cells. They look at upregulation.
so,
No, they don't do like a perturbation that much.
yeah, this is very much looking.
It's a
So
description.
yeah, it is quite descriptive. So I mean, if we're looking at well, so what is the most mechanistic paper that we have?
Coxiella. Is the Coxiella one the best from that perspective? Because
Uh,
they take plasmid out,
yeah, actually,
plasmid in.
yeah, I guess it is. Because again, they actually do an activity on the bacteria. Um,
I don't.
so
That interests you though at all?
uh, it, it's not especially interesting to me specifically. Um, but I mean,
No biggie.
because
No, it's fine.
uh, yeah, because there's the mechanistic aspects, but also trying to find a mechanistic paper that also has real like kind of value for like kind of
Not people deep in the field of Coxiella.
yeah. So
Yeah, I hear that. I hear that.
let me
Uh,
see.
I feel like we have to go back potentially for.
I mean, colistin. That's one that people because it's a potential antibiotic thing. It does do mechanistic stuff.
That's
But
true.
I feel like people will start thinking that eLife pays us, though. I mean, it's just that's fine.
I'm also tired of antibiotics at some level.
Yeah,
I think
I
that
understand that. I
antibiotics one. What about this is something that I think I did last. RNA thermosensors facilitate strep pneumoniae and Haemophilus influenza immune evasion.
I mean, I'd say that that's one where they've kind of already characterized the thermosensors, and all they do there is they just find the sequence. So it's not like
Yeah,
so.
that's right. It's not like, oh, we didn't know thermosensors were working here, but find the sequence like that's I guess that's very obscure. Mm.
So yeah, we're getting selective about papers now. We're turning into snobs. It's oh,
A little
there's an,
bit. I mean, yeah, I guess I'm just like I'm not sure what I'm craving to read and talk about.
I understand. We're at the rental store and we're going, oh, do we feel the need for a soap opera or does? Yeah, I understand that. Like,
Yeah, yeah, that's true. This is the wall of Netflix choices.
just like, oh, am I in the mood for this? It's a bit too. This documentary seems a bit too grim. Let's look at something more light.
I mean, that's like the trying to move that's why like I think we've added in other papers, not just SARS-CoV-2, right?
Yeah, to get more variety. Um,
Bashing it at all the time.
let's see. What other cool things? So, I mean, we had poor forming ESX proteins. That again, that we've been building off of that. Let me see. I mean, there's
I'm not against that, poor forming ESX proteins. Again, I think it's also I'm like trying to get to some yeah, I want to see more blots. I
more
want to see
blots.
West.
Let's
Ooh,
see.
they do cryo-EM in this pore forming one.
Yeah, they do. So I think that could we could do that.
The big paper, but
Let me see if I. Yeah, it is a pretty big paper, but I mean that hasn't stopped us before.
no,
Uh,
no, not at all.
so
I'm up for this if that caught your fancy at all.
let's. All right, so I'm gonna throw it up on the screen quickly so that people at home. So pore forming ESX proteins mediate toxin secretion by Mycobacterium tuberculosis. So this
Mhm.
So this paper has quite a lot of. It has microscopy. Some it has some cryo-EM as well. Right? Am I right? Am I
Yeah,
seeing this right? Yep.
I think so. Yeah, I think it is cryo-EM. I don't know what else would make that sort of picture.
Uh,
Guess we can just read the caption. Oh, the caption's so Kumasi stained. Yes, a EM session.
okay, cool. Yeah. So I just want to check that I've got the right paper on it. Okay, excellent. So yeah, we can do this one.
Yeah, I mean, like bacterial toxins are big, right? Like that's how bacteria make diseases in us. They inject a lot of things.
Yeah. So yeah, and it's still relatively recent. And it does some cool stuff. Um,
There are pictures.
yeah, there are pictures and
How do you know
tuberculosis.
when something is a pore-forming toxin?
Yeah. Okay. Yeah, we can do this one.
Okay, right. So join us next time where we're going to cover pore-forming ESX proteins mediate toxin secretion by Microbacterium tuberculosis.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with the hashtag. As you saw, we are responding to comments if they're left in our YouTube videos. We think peer review is a process which we can all participate in, and so we hope that you've had a good time listening to us ramble about microbiology today. If you have anything to add, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Bye.